All right, well, hello everyone. My name is Christian. I'm the pediatric fellow, for those of you who don't know me. And tonight I'm going to be talking about the applications of stem cells and regenerative medicine based approaches to airway reconstruction. And this is actually a particularly interesting field in that the clinical application was applied much more rapidly than the preclinical studies had been prepared for, and thus it was really thrust into the news and became kind of a controversial and interesting topic. So this was actually a featured story in Science in 2013 about the first stem cell-seeded bioengineered tracheal transplants that were done by Paolo Macchiarini and Martin Basal. At this point, they had already done more than a dozen of these transplants in which they took a artificial nanocomposite structure that was seeded with bone marrow-derived stem cells and transplanted it into patients who had either tumors or deficiencies in the trachea that required a large graft to replace, and for which there were no really other options. They claimed this really as the first example of a tissue-engineered organ that was transplanted, that the stem cells had engrafted and populated this structure that they implanted, and that this was a functional stem cell-derived trachea in a sense that just used the scaffold as a support means. But a lot of critics were not so sure about whether or not that was true, and I'll go into some of their descriptions, because there are certain things that you can answer in preclinical studies that it's hard to tell in humans, namely whether or not the cells and mucosa inside of the graft are actually from the stem cells that they say they put, or they're just the patient's cells. What was interesting was these quotes from Martin Basal when he was talking about the controversy when they got started back in 2008. And he acknowledges that, he says that we were 1 million miles away from where we thought we would need to know before trying this in people when they tried it. And also acknowledges actually that they really, he says we ran roughshod over the regulatory process, and with the permission, they basically were able to drive this through. It says it wasn't done with the highest possible standards, but it worked, and it changed the way that people think about regenerative medicine. So it's kind of a really bullish view of how they were able to get this to happen, but it was successful, and therefore, that seemed to justify getting it done in a quick way to them. But this actually just came into more attention in the public eye because there was a New York Times article yesterday, and this had been under investigation over the past year. But they actually, a board in Sweden has found that there was scientific misconduct committed in the publication of these studies. They published 3 different case reports in medical journals where there was omitted and falsified information about patient conditions. Most notably, in the Lancet paper in 2011, I'll show a little bit of their figures from that. They omitted some data, fabricated or falsified some other data about the postoperative state of patients, and they note specifically that they reported on the patient's condition after 5 months, but they found out that they had never actually even seen the patient after 5 months. So that was a complete fabrication of, basically, these reports were designed in such a way as to make the outcomes of the patients look much more favorable than they actually were. And there were several patients that actually had died who had received these transplants where there was no report about there was no autopsy done, there was no real report about what had actually happened. And so I think that, on that side, there's probably some valuable information that could have been learned about those cases that was just sort of totally brushed under the rug. And a lot of the cases were not published. They only published a few case reports, did not publish any series of their experience. So this is some of the pictures of what they did show in this 2011 Lancet report. And what you can see here is this is this composite graft, and it's made from a polymer that's very porous and allows for stem cells that they culture it with to engraft into these little niches and be in there. And they place this in a bioreactor prior to the transplantation. They've designed it, you can see in the cross-section, so that there's a more firm cast on the anterior wall and then this soft membranous portion in the back to be more like a trachea. This was the picture that they showed for 5 days post-operatively, where they said there's some evidence of early mucosalization, but I think it looks pretty rough at this point. And this was their picture. Yeah, this was actually a picture that they had showed. So as their evidence that this had successfully been engrafted in an organ, they took biopsies at different times after when they were doing bronchoscopies from the wall of the graft. And they showed that there was this mucosa that they saw on the wall of the graft. But what a lot of the critics in the scientific community said is, how do you know that the stem cells really are lining the graft and making the tissue? It could be that with or without inductive signals from the stem cells, that's just ingrowth into the graft, and that the graft may just be acting like a stent, and that over time some mucosa has grown in. If you do this experiment in an animal model, you can actually definitively answer that question, because you could, for example, take a stem cell seeded graft and have a parallel control where there was no stem cells and see if those ones have mucosa inside them as well. That would be evidence that the stem cells are not necessarily required to observe this. And then to definitively prove lineage, in an animal model, you can tag the stem cells that you're putting in with a GFP or some type of lineage tracer, and then you can just look at the mucosal biopsy and see, is it green? Is this coming from the stem cells really, or is this just from the donors?
That's a question. I'm hardly an excellent histologist. On the other hand, that doesn't look like any mucosa I've seen, unless it's extraordinarily inflamed. Is there anything that looks like mucosa in that?
Yeah, I think that this area here is kind of pseudostrate.
Is that?
This
it's
is
just
a little
dehydrated and delaminated there?
Yeah, it's not the best example. I think this, to me, looks
basal
like
membrane there?
basal membrane here.
I see, okay.
That's what I was seeing.
Certainly got a lot of cellular.
It does, yeah. Yeah. And then the other issue raised, because of these, the circumstances surrounding the death of some of the patients, which, again, were not really reported by the authors, but it sounded suspicious to people that they were admitted with large bleeds, and there was concern that maybe these stents were eroding into vessels. So the question is whether the scaffolds were really safe. And again, since there was no real studies done before these were tried, and then now the analysis of how the patients performed was suspect because of the reporting. We don't really know whether or not these are actually safe. And then the other question was durability over time. There's reports with this material that over the course of a year or more in a saline environment that it starts to soften and weaken. It was known that some of the patients who got these transplants had to have multiple stents placed at the anastomotic sites where there was granulation. But the question is whether or not the long-term performance of this scaffolding would be durable enough to function as a trachea over time. So there was a lot of questions, and these were all raised before the current ethical climate became even more questionable about the fabrication of data. And so I think it's interesting that all over the news there have been stories about the controversies with stem cells and regeneration of the trachea, and yet a lot of the preclinical work has yet to really be done. And so I kind of wanted to take a look back at, what does it really mean to say that we're going to regenerate something? And regenerate means, from the Latin, to create again, or creating again. And I think I wanted to stress the word again there, because I think a lot of times in the climate of tissue engineering, we get excited about the scaffolds and the technology, but we forget that we don't need to make something new. We don't need to figure out on our own how to make tissue. That we're actually doing something we're just trying to recapitulate what's already being done in development, and make something that has already made itself in the past. And I think the best example of this that I always found was really fascinating comes from the newt. And this is a limb bud in the newt. This will play. What does it say?
Yeah.
Is it your laptop?
No. It's
Probably
on here. I have it on, it won't.
It may need QuickTime Pro. Is it a WMD?
Yeah. So we don't have QuickTime on this
I
computer.
think
It's on the Mac.
it needs to be down. Are you working on the PC or the
On the
Mac?
Mac.
Probably it's a brand new Mac. It needs
Oh.
QuickTime Pro, not just
Okay.
QuickTime, in order to get a WMD. Do you have a lot of WMDs?
No, just two. But it was a good one. Yeah.
This is Bill Gates' revenge. They don't have the Windows Media Player.
What about Flip for Mac?
Or VLC.
It won't.
Or VLC, yeah.
Sure. All right. So basically, this was a movie clip that was showing sort of a time-lapse video of the limb bud of the newt that was generating a new limb. And what happens, and then they show a dramatization of how the stem cells are able to signal to regenerate a limb that has proper patterning after the limb is actually cut. So what's interesting is that this process occurs, and it occurs in a setting of wound healing. And that is that after the fresh cut is formed, the wound first closes down, and it has all of the pro-inflammatory environment and the necessary contraction to close the wound. And from that environment, the developmental process resumes. And actually, the same pattern of inductive factors and progenitors is able to form a tissue that contains muscle and bone and nerves and epithelium, and it's all patterned properly, including with 4 digits in the right orientation and with function. And so the fact that this happens in nature in vertebrates on a consistent and reliable basis led me to think about, so how do you define regeneration? And I think that maybe a better definition of it is really that you want to recapitulate aspects of normal development in a setting of wound healing. That is, you basically want to take a wound and make it the way it was before, in the right way. And so I think that if we can figure out how to understand normal development and be able to influence the environment of wound healing to proceed closer to normal development, then we can potentially influence and have an impact of regeneration in a natural way. And so in the setting of airway stenosis, there's a couple of applications I think that are in broad categories that could be really useful in terms of regeneration approach. The first major category is just on improved wound healing, prevention of granulation tissue and scar formation. There are a number of interventions that we can do both endoscopic and open in airway surgery that initially look great and can solve the problem, but over time, because of poor wound healing, formation of granulation tissue, and then new scar within the lumen of the airway, we get repeat narrowing and then failure. And so I think that there's a need to understand from some basic animal models and studies what causes airway stenosis to form on a molecular level, and then understanding the role and the behavior of stem cells within both normal development, we need to understand that more, and then in injury models where there's a wound that needs to heal, what are the stem cells doing, and how can we try to recapitulate more of a normal developmental process and less granulation and scar tissue formation. And then the other more broad goal, which is sort of what these transplants had jumped ahead into, is in terms of tissue engineering, designing and building grafts that can help to repair large defects and that offer safe and stable reconstruction options. And so I was looking at what animal models have been around and what the advantages and disadvantages of them are, and what would be the best model to really study wound healing and regeneration. And the rabbit model has been used and offers some advantages. In this left panel, what you're seeing are direct laryngoscopy views, and endoscopy is one of the advantages in the New Zealand white rabbit, that it's large enough that you can perform endoscopies. And in this model, they have looked at wounds formed by either a CO2 laser or silver nitrate burn, and you get this subglottic scarring. And then it's a model for surgical techniques as well. They were able to excise circumferentially around these stenosis, and then they observed that by day 14 they had some re-narrowing forming more significantly in the silver nitrate. But so this is a good and useful surgical model that offers endoscopy. The other really recent study that was done with the rabbit model has the advantage of that it's a intubation-based injury, which is really analogous to how a lot of stenosis is thought to form in patients. And what they did is basically intubated with a size 4 tube, 4 millimeter tube, the rabbits for either 2 hours, 4 hours, or 6 hours, and then extubated them and let them basically live for another 12 weeks and examined what happened. And what they saw was, especially in the 6 hours and somewhat in the 4 hours, they saw a remarkable formation of this thickened stenosis over time. In fact, several of the rabbits at the 6-hour time point did not survive to make it to the analysis at 12 weeks because of airway stenosis and collapse and events. The rabbit, one of the disadvantages of the rabbit, and I'll get to this more a little bit later, though, is that beyond the surgical aspects of it, there's limits as far as genetic types of experiments that you can do, which are more possible in the mouse. And because of that reason, people have tried to do models in the mouse. And the most common one that's published is a model of injury in the subglottis of one mouse. And what you see here in the top left is this is dissecting out to expose the airway. They make a small tracheostomy, and then with a monopolar cautery aimed to the posterior cricoid mucosa, they make a burn on the posterior cricoid. Then this whole area, including the first 3 tracheal rings, is dissected out in what they call the laryngotracheal complex, and this is then transplanted into another mouse in a subcutaneous pocket in the back, and left for a various period of time between 1 and 4 weeks in this study. And this is showing it at the time of harvest. You see that they've opened it up. There's all of this inflammation and some ingrowth of vessels into this, and then they've dissected it out here. With their analysis, they look at mice which had no injury performed as a control, and the ones which had the cautery injury performed. And what they see is that over time, this is an example of staining at the 3-week time point. And in the experimental, they say that there's this thickening of the submucosal layer known as the lamina propria, with the proliferation of mucus glands and fibrosis in that layer. And you'll see that on this graph here, that compared to the control, already at 1 and 2 weeks, there's a trend towards an increase in thickness, which becomes statistically significant at 3 and 4 weeks. And probably is not as significant here because there's a lot of variability early on in the control as well. And this model was used by other groups for some interesting studies. I think this one is particularly interesting because it takes advantage of this heterotopic transplantation between 2 animals to look at the intrinsic aspects versus the systemic aspects of what could be causing the stenosis to form. And so what they did is they had 2 groups of donor mice, one which was treated with mechanical and chemical injury and control, and transplanted into a SCID mouse, which is an immunodeficient mouse that lacks a systemic inflammatory response, does not have any cytokines or cell-based immunity. And then the other group, again with the different types of injury and control, was just transplanted into a wild-type mouse. And then they looked at whether or not the recipient, their systemic inflammatory response was important for the formation of stenosis. And they found that actually it was completely essential. When they transplanted these wounded tracheas into the immunodeficient mouse, they did not see any increase in the lamina propria thickness. It behaved just like a control. And so the increase in thickness was completely, in this study, was completely dependent on having a recipient immune system response, which is an interesting point. But I think one of the caveats or concerns with this is that it also brings up the really artificial nature of this model, in that you're taking an organ and placing it in a completely new environment, and that this subcutaneous transplant environment does not have any air epithelial interface, would not be exposed to oropharyngeal bacteria or biofilms or signals, also needs to become completely neo-revascularized because you're just putting it into a pocket, and that would require a whole different inflammatory response and mobilization of vessels, all of which really has nothing to do with airway stenosis. And then the other thing is, although these mice are from the same genetic background and typically don't have rejection phenomena, there still could be some type of a foreign body inflammatory response, because you're putting an object in a place that it doesn't need to be, and it needs to heal. And like we mentioned, it needs revascularization. And so a lot of that inflammatory response it really has nothing to do with an endogenous situation, and it could be clouding the picture in this model. So in terms of improving an animal model to study mechanisms of stenosis formation, I think there's sort of two important criteria which would be great to have. The first is that you get stenosis formation in a native airway environment. And that's really what the rabbit models have provided, that they're able to look at a living animal with airway stenosis and not an ectopic transplanted system. And then the other thing would really be to try to do this in the mouse, where you can take advantage of genetic tools, different knockouts and knock-ins and tracers, to really answer some of the questions about what factors are involved in this process, to look at the behavior of stem cells with different lineage tracers. There's really a lot of things that you can do if you can do this in the mouse. And so the idea that I had really was that a tracheostomy mouse model, if it was feasible, could satisfy these points. And the advantage of the tracheostomy, an indwelling tracheostomy, would be that the tube which you're using to cause pressure and irritation and stenosis could also stent the airway open so that the mouse would not die from obstruction, which I assume was probably the reason why some of the tactics that were noted in rabbit have never really been done in mouse, because the airway is so small that if you make a wound, it's likely that the mouse will just steno shut. So I looked around and found that there were several reports about, not several, but some reports about tracheostomy in mice. Probably the most common was in the pulmonary literature, where they would use a tracheostomy primarily as a vehicle of delivering noxious gases to the lung in a direct way. And in those experiments, they would basically make a tracheostomy, inject something, and then take it out, and then let the mouse recover. And then I found this paper, which actually gave a really nice pictorial description of how they did the procedure. And this was from the neuroscience literature, where they basically were using a tracheostomy during a neurophysiological recording experiment to keep the mouse alive for a longer period of time on urethane anesthesia, and where they had trouble with oral intubations. And what they do is basically used a angiocath tip with a flange on it, and after dissecting out and exposing the trachea, they use a needle in sort of a Seldinger technique to push this tracheostomy tube into place and then secure it. And so I tried this. After seeing those pictures, I had to try it. And this probably won't play then too. We'll see. Oh, this one's playing, but it's sideways. But basically, this is the very first mouse that I tried this on. And it's really warped and blurry. But you can see that he's moving. So I just wanted to show you that that is a live mouse with a tracheostomy tube in, who is relatively, this is immediately post-op, but relatively not in distress. He had a happy face if you saw it on. But basically, so then going from this, what I wanted to do was more of a formal pilot study to look at what the effects were of a tracheostomy on stenosis and do some histology. And so I took 12 mice and they underwent tracheostomy using a technique similar to this, where we basically pre-set up all of these angiocaths with a little silastic piece on there that we made from actually cutting up Doyle splints into little rectangles. And these were preloaded. And then after the airway was exposed in the midline, we were able to pass this tube under direct visualization through the tracheal rings. And then this was all secured down and closed. And these mice were then allowed to go back in their cages. And this is, so unlike the people that have, did the tracheal transplants in humans, I'm going to report that there were some complications here. And I had 4 intraoperative deaths. Some of that was related to the anesthesia, and interestingly, immediately there were some cases where immediately after putting the tube in, the mouse became really under distress and was breathing really rapidly, and if you pull the tube out, they would calm down, and then sometimes you could replace it and they would be fine. But failing to recognize that early on, some of them I would just put the tube in and then they just arrested during the procedure. So it was like some type of a stress response, not entirely well understood. I should mention that for the anesthesia for these, it's basically an awake tracheostomy done with ketamine and xylazine anesthesia. So we just an intraperitoneal injection of the ketamine. The mouse is moving and breathing spontaneously with a little twitching, which is why we have to tape them down to secure them to do the procedure. There were 3 of the mice that were found on post-op days 1 and 2 in their cages that were dead, and on examination of them, they all had some secretions and blood in the tracheostomy tube. I don't know exactly what happened, whether that was a consequence or cause of death as far as mucus plugging, but that's also one of the definite possibilities in having a small tube where you're not suctioning or humidifying and just letting the mouse walk around, that it could plug. So it's actually quite remarkable that 5 out of the 12 mice in this initial trial did survive to 6 days. I ended up stopping the trial at 6 days. I was going to just do a week to be round, but one of the mice was looking a little in distress, and I didn't want to lose any more numbers, so I just took the whole colony at 6 days. And decided that way we'd be able to get some type of statistics on this for an initial pilot. And what I ended up doing then was to analyze the histology of the subglottis and trachea from the 5 mice that survived to 6 days with the tracheostomy versus 3 control mice. I used a lower number of control mice mainly because there was very little variability in those mice, so I didn't feel like I needed more in the initial comparison. The tracheostomy tubes also, I should mention, were placed as high as I could because I wanted to have more length of the tube present in the airway, both from the point of view of securing it because the mice do tend to pull on the tube. And so I was concerned that if I left too short of a piece in, that the mice would easily decannulate. But I also didn't want them to main stem, so I put as long of I placed the tube in as high as I could. And therefore also the advantage would be that the tube would be up near the cricoid and by the subglottis. It was just sort of right under it. And this is sort of some pictures of the histology of what we saw. This one, I think I was able to get it right around where the anterior wall injury was, and there's some coagulum there inside. But what you see basically, we performed an analysis similar to what was done in the laryngotracheal complex papers, where we measured this thickness from the basement membrane down to the cartilage of this lamina propria layer, and saw that there was a statistically significant increase in the thickness in the experimental mice at these levels around the tracheostomy. Between that and where the typical thickness would be at similar levels in the control. And here you can see as markers that you do see some of the, I think what's the posterior part of the cricoid just where this is coming in, the esophagus is posterior, and you've got thyroid in both sides here as well as part of the histology. So this model is able to show evidence of histological evidence of airway stenosis formation at this 6-day time point in the airway. We also took a look at some molecular markers. Vimentin is expressed in fibroblasts in this lamina propria layer, and I took a look at proliferation using a Ki67 marker. And proliferation in these Vimentin positive cells is really very low. The number was about 0.5% or so of cells in the control. But there is a boost up to about 22% or so of dividing cells in this fibroblastic layer. And so this is potentially indicative of a fibrotic response, and it's something that I want to be able to take a look at with more molecular markers and see what's going on in that layer as far as proliferation of fibroblasts explaining this increased thickening. So there's a number of things I want to do with future directions in this model. One is to attempt to decrease the mortality through technical improvement and experience with the surgical procedure, and to evaluate the effects, if possible, of a longer duration of tracheostomy. If we can get improved survival, then we can start to let them go for 2-3 weeks and see what effects that has in terms of maybe making more of a mature scar. Also, and probably most importantly, since that's why I think the mouse model is useful, to perform tracheostomy in genetically modified mice versus controlled mice, and then compare the degree of stenosis formation. And a couple of mouse lines that we have ready to go are actually kind of interesting. TGF-beta signaling pathway is important in fibroblast proliferation and repressing epithelial growth. And so, and it does have effects on both fibroblasts and epithelial migration in wound healing. And so we can specifically knock out TGF-beta signaling via the common mediator of SMAD4 in epithelial cells in an inducible manner by giving doxycycline, and we can, in different mice with a fibroblast promoter, we can specifically knock out TGF-beta signaling in the fibroblasts. And then the thinking is that we may be able to actually functionally pin down what cell population and what gene is required for stenosis formation. And there's going to be multiple factors, but it's these types of in vivo functional studies that really haven't been done. A lot of what has been done has been expression data in human samples, but that doesn't really tell you what factors are actually causing these processes to happen. And it's really the genetic experiments that tell you what's happening. And the other thing I wanted to do was to examine stem cell behavior in this type of an injury model and see, looking at markers of the epithelial stem cells and mesenchymal stem cells, what's actually happening in driving this process in wound healing. Because I think that might be where, if we understand the genetics and the cell populations at work, where we can modify it. And if there's problems with the survival and practical concerns, the alternate backup system is to use the established laryngotracheal complex model to do these types of studies where the survival should not be an issue. But I would prefer to do it in a more natural setting. The other thing which can be done, and I think is a good idea in terms of doing functional studies on human samples, is to develop an explant culture system where you can look at explanted tissue, and this has been done a lot in embryology, and I'll show a little bit of that, but where you can actually do experiments on tissues that are growing for a short period of time in an explant culture. And so I wanted to talk a little bit now, in shifting gears, about some of the recent and exciting work on what is known about stem cells and development of both the epithelium and the cartilage in the trachea. And the basal cells of the tracheal epithelium act as the stem cells. And a kind of a neat experiment for people in developmental biology was this paper where they showed that if you plate basal cells that are isolated at a clonal density, they'll actually form what they've called tracheospheres. And what these are these little bubbles that basically have at their outer part cells that express P63, which is a marker of the basal cell stem cells. And then these will grow and gradually produce differentiated progeny, which move towards the center. You can see that here with the keratin 8, which is a marker of the progenitor cells. And then if you wait, this is what they see at 9 days. If you wait out to 20 days, what they actually see on the H&E staining is cilia forming on the inner surface and acetylated tubulin, which is a marker of ciliated cells. And so they actually can get differentiated progeny forming from these single cell colonies. And they grow in this fashion where they face a lumen, and then the differentiated progeny move toward the center. So it's like little mini balls of trachea epithelium. And this is actually interesting because this was the traditional model of how the stem cells would generate the different types of epithelial cells in this pseudostratified columnar epithelium, where you had a uniform population of basal cells, and then it would give rise to multipotent progenitors that would respond to signals like Notch that would tell it either to become a secretory cell or a ciliated cell. And that model actually is being revised now because of a recent paper in Cell Stem Cell, where they've showed that in response to sulfur dioxide injury, where you take away all of the luminal cells and have just a basal cell layer, and then this basal cell layer will regenerate the epithelium. But they were able to look at this population, and what they see is that there's a very small percentage of cells that express this marker CMIB and another one that express this Notch2 intercellular domain. And in response to the injury, after about 24 hours, what you see is that there's a segregation of these different basal cell populations, and you actually start to develop significant proportion of the basal cells that are expressing these markers. And that this basal cell segregation happens before the commitment to these multipotent progenitors. And so they've changed the model now to basically show that there's a heterogeneity of the basal cells, and that now they group it into really three populations, which are the P63 only expressing the so-called stem basal cell, and then there are these other, and that actually at the resting state is the majority of cells in gray here. And then there's the Notch2 intercellular domain expressing P63 also expressing basal cell, and then there's the CMEB population. And that these cells appear to be on a pre-specified trajectory already as a basal cell. So it's a little bit of an interesting thing that there's more of a complexity in what we're previously calling as stem cells, and that they can actually undergo this segregation process before they start to form progenitors. And what they also show in terms of the role of Notch signaling is that it acts at that level, actually. And so what you'll see is if you do this in a setting of a knockout mouse where the Notch pathway is inhibited, then you get an impairment of this segregation, and in that mouse you will just make ciliated cells. You cannot have, and you'll see only the CMEB expression in the P63 pathway. So it's really a switch that happens in the stem cells, not later on. So that's kind of an interesting new finding that just came out this year, and it sort of changes the way that we might think about the dynamics of how different progenitors are formed. And so shifting gears a little bit to talk about cartilage. Cartilage is another very important structure in tracheal development. And in a wild-type mouse here, you see the cartilage along the anterior part of the trachea, and then the trachea and esophagus here. And then they show this from different angles just so you can see the cartilage from a side view, from the front, and then from the back where you've got your membranous portion here. And sonic hedgehog is a morphogen that's involved in a lot of different organs development, and it's an essential factor for tracheal cartilage. In the sonic hedgehog knockout, you basically have very little staining with this Alcyon blue here, and really no expression of cartilage. You also have just a common tracheal esophagus that doesn't really separate out. And then This is also the SOX9, or the sonic hedgehog controls the expression pattern of SOX9, which is a pro-cartilaginous factor. And you see that in the sonic hedgehog knockout, you have a disorganized expression. There's some ectopic SOX9 dorsally, and you lose this nice ring, which is helping the cartilage to form. The other thing that you don't see here, but in a segmented view, the SOX9 also has an expression that mirrors exactly these cartilages. That is, in the rostrocaudal axis, it has these intensities that are patterned out. And that's also dependent on the sonic hedgehog expression. What's interesting over here, this is an example of an explant system where they've taken these, they've explanted the developing tracheoesophagus tube area at around E10.5 and then let it grow for 8 days in culture. And in a wild type setting, these here, the cyclopamine is a sonic hedgehog inhibitor, and this is an analogous molecule that doesn't do anything to sonic hedgehog that they use as a control. So normally when they do this explant, you get a ventral formation of cartilage here, anterior to the trachea in a wild type explant. If you treat with this inhibitor of sonic hedgehog, then you fail to get the formation of the cartilage. This is a nice experiment here because in the sonic hedgehog knockout, what they see is normally if you grow it in this system, you don't get any cartilage formation. But if you add back sonic hedgehog to the medium, it rescues the formation of cartilage. So that is a really nice, clean type of experiment that you can do in an animal model or in an explant, where you can show definitively that the sonic hedgehog is necessary and sufficient for this cartilage to form by gain of loss of function experiments and rescuing the phenotype. And there's other genes that have been studied and published with phenotypes that are interesting regarding tracheal cartilage and patterning. Fibroblast growth factor 10 is an important gene for proper tracheal cartilage patterning. In the knockout, what you see is this really shortened trachea with aborted rings that looks really abnormal. And in this lateral view, you see it has no structural integrity because of that. And this is an interesting thing where they looked at the heterozygotes for so the sonic hedgehog mutant, like we showed in the last slide, has a severe phenotype like the FGF10. But both of those, when in the heterozygous state, they resemble wild type more or less. They have nice tracheal rings forming here. And the double heterozygote, when they cross the two, they start getting this unusual phenotype where you get these abnormally branched formation of tracheal rings and some of these aborted rings here that fail to come around completely. And so that shows that these pathways probably interact at some point and are important for the patterning of the trachea. This phenotype is not the same severity as either of the homozygotes, but it's more severe than the heterozygotes. So there's some type of an additive effect going on there. The other really interesting thing I found in this paper was actually that they did a time course of induction of overexpression of FGF10. And what they found is if they overexpress it in this time window from E10.5 to 11.5, it didn't do anything. And similarly, if they overexpress it later at 13.5 to 15.5, it didn't do anything. But during this critical time window, if they overexpress it, what you get is a total loss of this patterning of the tracheal cartilages, and you get this fusion along the pathway from rostrocaudal, kind of like a tracheal cartilage sleeve. And this is actually also interesting because FGF10 is the ligand for FGFR2, which is the gene mutated in Apert syndrome, where you sometimes see these tracheal cartilage sleeves. And that was something that they just uncovered. So it's just an interesting phenotype, and I think that these types of analysis of the normal development and mutations can really help us show what factors are important in formation of tracheal cartilages. And I think these pictures really help illustrate how intricate this process is, and how the patterning is really something that is well controlled, and that you can get these phenotypes that have these unusual patterns. And I think there's going to be some interesting downstream targets of these genes that will explain this all more in the future. And then with regards to cartilage, there's interesting work being done as far as trying to regenerate, if you will, like a tracheal structure in a way that would be scaffold-free, that would not involve needing to have any type of permanent framework. In this experiment, what they did is they seed human mesenchymal stem cells either with or without TGF-beta microspheres to try to help with cartilage promotion and formation. And they grow them in this disc array. So this is seen from the side view and from the top view. So you get a formation of a little cartilage ring. And then they allow this to happen for 2 days. And then they stack these rings that they get either as a 3-ring tube or as a 6-ring tube along these little silicone tubes. And then they have them grow for 22 days total in culture. And what they see in both cases, they got these little cartilage tissue rings, and then these 6-ring tubes actually have the same type of size, the way they designed it, more or less as a rat trachea. And so, of course, the main difference is that these are circumferential rings. The other difference that they really show here is that their rings have a much thicker thickness than the rat trachea cartilage, and the overall tube's outer diameter is also larger. That said, and this is nice because it's a bit of honesty, because what they go on to do is look at the mechanical properties of these cartilage tubes, and they see that these structures behave favorably compared to the rat trachea. They're able to bear a good amount of load before failing, and they looked at the load at 80% collapse. And really in all categories they tend to, and the tensile stress force, the recoil, the percentage of recoil after it's completely collapsed. And with all these types of parameters, they tend to perform well. But I think one of the caveats with this is that these are thicker, more robust rings as well. So it does sort of remain to be seen, what the mechanical properties are. But I think overall, this is an encouraging thing that you can get cartilage that acutely at least has some type of performance in terms of strength that's generated and could potentially provide some hope for scaffold-free development of cartilage rings. But that's still something that really needs to be studied a lot more. And so in conclusion, really, I just wanted to say that, as far as regenerative approaches to airway construction or to airway reconstruction, I think that, we really need to learn a lot more about the molecular mechanisms that underlie stenosis formation, and that one of those aspects is also understanding the role of stem cells and progenitor cells, and their role in both airway healing in wound models and also in normal development. And I think that the overall theme of trying to recapitulate aspects of development in a wound healing setting is really what the regenerative approach is all about. And so they kind of go hand in hand in terms of understanding both the inflammatory mechanisms of the wound healing environment and normal development, and then trying to see if we can modulate that. And that goes to the third point here, really, which is, can a better understanding of these factors lead to new therapeutic improvements, both in terms of reconstruction, as far as tissue engineering, and as far as preventing stenosis recurrence after surgery by just simply improving wound healing by producing drugs or other factors that can specifically augment the situation so that we have less scarring. Okay, thank you. Yeah.
Is it easy to get enough mesenchymal stem cells to, see framework? Like, are they plentiful in the peripheral blood, or are they? You have to, apheresis the entire blood volume multiple times to get them out. Like,
I mean, that was they were bone marrow derived in that study, and I think they had to be amplified up with a protocol. I don't think it's that easy to just get that much. Yeah.
So the mice that live with your angiocath traits, how do you maintain that? Because in the New Zealand rabbit, you can't leave those things for 5 minutes without their little trach tubes clogging. That's one of the downsides of the trach device. So that's incredible. If you just walked out of the room, 5 of those things survive.
Right, and so actually there were some, we did check on them a couple times a day. I know that's not that frequent, and there were a couple of times where I thought that they were plugging and actually figured out a little way of how to suction them. because there's no suction tube that's small enough, but you take a smaller angiocath and hook it up to a syringe and just pull out. That mouse that I did that on didn't make it though. But
No double lumen tubes.
right.
The level of, and the second question I have on that project though, is the level of stenosis that you're looking at histologically, is that from the injury from the trach or is that proximal?
It's
And
a little
what's
bit.
the utility with regard to reflecting a stenosis model?
Well, I think it's at the, it's like a suprastomal level basically. It's kind of right at the bottom of the cricoid where the trach is going in.
Gotcha.
I think that's where it is.
Gotcha.
Yeah.
Christian, how many of the virtual, I forget his first guy's name.
Machiavelli, yeah.
How many of their did they actually create with the scaffolding, and how many survived?
They've done more than a dozen. It's a little nebulous because they never published a series. They published 3 different case reports. The very first one they did was with a decellularized matrix that was seeded with stem cells from a cadaveric trachea. I think the one...
Spain?
Yeah, in Spain. And I think the first one that they did with the scaffold was that one in 2011 that they published in Lancet. But the numbers, it's kind of hard to get your hands on the true numbers because it's mainly just in popular media. And they tend to focus on the dramatic aspects and not the details of the,
What was the status of these patients that they got approval for to use? These people on
They were
death's
patients,
door,
yeah,
or-
basically.
Yeah.
They were all, I think that's how they were able to push it through. They really did not have any other option on the table. And some of them were cancer patients where they had a large obstructive tumor that was and it was involving the bronchi in such a way they didn't think it was resectable. Like you notice on that scaffold that they showed it had the bronchi on it as well. So for some large defects where they really didn't have another surgical option, that was when they used it. So, and in that respect, I think it's on the one hand, I think it's quite reasonable that there were some patients that failed. I mean, these were sick patients. I think the real problem is not so much that there was failure; it's that there was shady reporting. I mean, I think we could have learned a lot from those failures if we had followed up.
It's an interesting comment about really the last century of surgical innovation. Is that well, it is true that the lack of FDA types of drug controls with pre-market and observations and enormous data sets and $1 billion to bring a drug to market, were it applied to surgical innovation, would have a huge damper. I'm not recommending that. At the same time, there are many people that just try something out. And the otolaryngology literature is full of people reporting new operations, for vertigo, for example, and they'll report 95% success. And then 2 years later, you don't hear anything more about it. And usually the same people then introduce a new operation that is also 95% successful, never mentioning they abandoned the former. So, I think this is an example of lack of rigor. They have every reason that they should be reporting every case in detail with the full details of it, and it's very concerning that they didn't. Yeah. And I'm not in favor of an FDA-like thing, but I do think that we in surgery need to be thorough about reporting our data and in a sequential way and being upfront about it. I mean, new innovations in very ill patients oftentimes don't work. Right. Going to the media first with case and another case and another case to hit the Huffington Post rather than, having integrity in Lancet is a serious problem, and it's a temptation we all face. We all want great results. But I just urge all of us to be thoughtful about being rigorously honest and thorough in our data presentations, just as you did with your dead mice.
Right.
Thank you.